Bismillah ar-Rahman ar-Rahim. The title of our lecture is Immunity to Infectious Disease and Vaccines. First, we have the viral infection. The first line of defense mechanism in viral infection is innate immune response, which include recognition of the pathogen associated with molecular pattern, including the expression of the interferon alpha and beta, and the second, activation of the NK cells. And if this innate immunity fails to eradicate the viral infection, then the humoral immune response or the adaptive immunity will react. And this includes the humoral immunity. And the humoral immunity is consisted from the secretory IgA, which is present in the mucous membrane and in the secretion, and this blocks the binding of the virus. And the another antibody is the IgG and the IgM, and the IgA block the fusion of the viral envelope with the host cell. And in the primary immune response, the IgM, while in the secondary immune response, the IgG. And these two antibodies block the viral infusion with the host cell. And the IgG and the IgM enhance phagocytosis and activate the complement. And also the IgM had another function that agglutinate the viral particles. And the adaptive immunity have also another part, which is the cell-mediated immunity. It had a play, and this cell-mediated immunity play an important role in the eradication of the viral infection. And this includes the Th1 CD4 positive that secrete cytokines like interleukin-2, tumor necrosis factor, and interferon gamma. And also the cytotoxic cells play an important role in the viral eradication, which secrete interferon delta that has direct antiviral activity. And cytotoxic T cell kill virus infected cell, and also the NK cells and macrophage kill viral infected cells by the antibody dependent cell mediated cytotoxicity. This is the immune response against the viral infection, and the virus also has had another mechanism that evades the host defense mechanism, and this includes the virus encode proteins that interfere with the innate and the adaptive immunity, and also inhibit the action of the interferon like hepatitis C virus. Also, the viral inhibit viral antigen presentation by the host, and for best example is herpes simplex virus. The another evade mechanism reduce surface expression of class 1 MHC molecule on the cell surface, and best example cytomegalovirus. Some viruses secrete proteins that inhibit binding to C3b, inhibiting classical and alternative pathway of the complement, and best example the herpes simplex virus. Some viruses had another mechanism of evasion, which is change their surface antigen, like influenza virus, which is called antigenic shift and antigenic drift, and also the rhinovirus and the HIV virus, human immunodeficiency virus. And here we explain what's the antigenic drift and what's the antigenic shift. Antigenic drift, which is a series of spontaneous point mutation that occur gradually, resulting in minor change in heme agglutinin and neuraminidase. While antigenic shift can be defined as the structure of the heme agglutinin and or neuraminidase are different from the virus present in preceding year, and emergence of new subtype of influenza due to reassortment of entire single-stranded RNA between the human and animal virion infected the same cell. And this can be explained in this figure. This is the bird which is infected with influenza A strain virus, and this is the human which is infected with human influenza A strain virus. and this is usually occurred in the village or in the area where uh, were breeding animals. And here the pigs, and in the pigs, this pig infected with the bird virus and human virus. And the virus passed from the bird to intermediate host. So the pigs is the intermediate host. And here in the intermediate host, there is infection of the two strain, human strain and the bird strain. And here there is in this intermediate host, so there is reassortment of the genes, and genes from each strain mixed to form a new virus strain. So there is emergence of the new strain that contain genes, some from the bird and some from the human, and this leads to emergence of the new strain spreads from the intermediate host to a human, and this leads to new strain evolve further and spread from the person to person. So this is called the antigenic shift. So there is emergence and there is a complete change in the heme agglutinin and or neuraminidase receptors on the cell surface of influenza virus. While in the antigenic drift, there is a point mutation that occur, which is result in minor changes in the heme agglutinin or neuraminidase, while in the shift that there is complete change in these receptors, heme agglutinin or neuraminidase, which is lead to emergence of new strain of the influenza virus. There is which is called the original antigenic sin, that the body immune system utilizes immunological memory cell based on previous infection when a second slightly different version of the pathogen, like virus or bacteria, is encountered. And this leaves the immune system trapped by the first response it has made to each antigen and unable to mount potentially more effective responses during subsequent infection. And this phenomena can be found in the influenza virus, dengue fever, and HIV virus. And here in this regard can explain the above. And this is the adaptive immune system. And the adaptive immune system produced antibodies against the virus. And here this is the virus or infectious agent. And this is the antigen, the blue one. And this is the antigen, the red one. So the body will form antibodies against these antigens, blue one, and against these antigens, the red one. And this antigen, the red one, is recessive, which is the number of the antigens on this infectious agent little, only 2 antigens, while the major antigen, which is the blue one, 6, 1, 2, 3, 4, 5, 6. So the body formed antibodies against a large amount of antibodies against this blue one. Sorry. And a small amount of antibodies against this recessive antigen. And this leads to, while this blue one is the dominant antigen, and this leads to strong immunity. While when there is infection with the mutated infectious antigen, this virus or and when there is a mutation in this virus or bacteria, and this leads to a change in the antigen. So the number of the recessive antigen, which is the red one, increase in its number, while the dominant antigen, which is the blue one, decrease in its number. So the amount of the antibodies produced by the body is against this red one is small, while there is large number of antibodies against this blue antigens. So this end in the weak immunity. So this is called the what's called original antigenic sin. So the immune system will be trapped and did not distinguish between these two viruses or two pathogens, because this pathogen will be mutated and change in its surface antigen. Now we have coronavirus disease 19.